so we can still access the information, right? So we don't like wrap it like a spool of thread, 100 times around the spool. We wrap it just twice so that we can still read the information around there. But that doesn't mean that information is totally accessible all the time, because if you bind it in such a way, maybe like, hold it tighter to the spool, then you can't really sort of get at those particular reactive groups that need to be written by particular transcription factors. So by adding acetyl groups, you appear to loosen the attraction in this particular area. So I believe these results has been simulated. And so they loosen up a little bit, which probably then is going to allow greater accessibility of the information in those particular areas, which again appears to be critical in terms of the ability to mount the entire complex at this promoter. So now notice that what we've done is we now have a dovetaling here between our aspects of regulation of transcription by means of transcription factors in the classic way, and also compaction and positioning, which is related then to things such as epigenetic marks. So here we have a transcriptional coactivator that is going to add transcriptional marks and therefore change the ability to bind other transcription factors and so on as well. So we have like, again, another linkage. It's like, it's not just that we have epigenetic stop and then think about transcription factors, but here we see a linkage between transcription factors, transcriptional coactivators, and epigenetics. We're changing epigenetic markings to change the probability that we will transcribe a particular gene. So again, you can see the importance and probably why this is like a key player in terms of this promoter, KLF11 binding site, is because it's very important in binding P300, and P300 is very important in the assembly of the complex, and not only, of course, just the assembly of other coactivators and transcription factors, but also modification of the chromatin. Modification of the chromatin to make the information on that chromatin more accessible, and therefore further facilitating the formation of the complex at the promoter that's going to be used in the regulation of transcription.
And acetylation is opposite of methylation, right? Where we methylate groups in order to condense them to be.
Yeah, and I wouldn't say that dogmatically. I mean, what I would say is, yeah, at least in some of the patterns, that would be a generalized pattern that we see. That if you increase acetylation, then you tend to decrease convection and increase the ability to transcribe genes in those areas. So yeah, it tends to go in the opposite direction, but it's complex and it's not understood. We got all these other marks, and it's also going to depend on like where exactly do you put the marks, and those things have not yet been intended yet either. But in a real crude sense, in the crudest sense, if you will, that I gave you before, they do go in opposite directions. So if you methylate cytosines, then that tends to shut down promoters. If you acetylate promoter releasing, then that tends to activate chromatin. In a real crude sense, indeed, that is the case. But of course, there's lots of other markers going on, and the precise nature of the markers, at least, as a way to explain the whole concept of how we're using these markers to change that kind of whole.
And one more question, what do you exactly mean by, because I know it says it's poorly understood here for non-histone chromosomal proteins. So as in P300, does it mean that the DNA strand is using protein scaffolding if it doesn't have any histones at all?
Actually, that's sort of a different topic. So when you talk about a scaffold for a chromosome, what we're really talking about is what's left over when we disrupt the whole chromosome and see what's left and how those are organizing. So you were reviewing, right? You're coming from earlier, right?
Yeah.
Let's talk about this mess. So this is sort of where we had talked earlier, and what we've done here in the series of pictures I used, we sort of disrupted the binding of the DNA to the protein. So we had reagents that partially sort of disrupted the proteins, the chromatin. And this would be, what does it look like? Chromosome 2 maybe, or something like that. And it's been disrupted. And so this is a DNA all spewing out, or spewed way over here. But then when you do that, you see there's still some protein framework that's left, like the residue, the skeleton of it, kind of thing. And so there's something left here. That's the scaffold of the chromosome, if you will, which is pretty much totally not understood. Like zero, like virtually zero information on this. We don't know what this is, and of course, this is just like, you know, the skeleton left over and it's been disrupted. So to disrupt it like this, we have these proteins. And then we're just seeing, but look, even though the DNA is spewed all over, there's still a matrix, there's still some kind of a framework here, some kind of scaffolding. And so like right now I'm thinking about like say Cholla cactus, and you see like the dead stems, and you see like the skeleton that's left, and that's sort of what we're seeing here. This skeleton that's left here, that's the chromosome scaffolding. So when I talk about a scaffolding, I'm sorry?
What did you put?
We don't know. I mean, what's left here is protein. Now, does that mean that there's not RNA involved? No, there might be RNA involved, but until people look for RNA, they're not going to know. We know, for example, very clearly that in the formation of our bodies, RNA is a key player. And there's particular RNAs, and there's something called exis, for example, that is involved in again, another topic would be great to study that would like, we take like a whole lecture to talk about our body formation, but in a nutshell, what happens is that we start accumulating RNAs, and we transcribe particular areas of the X chromosome. Those produce what are called long non-coding RNAs. Those long non-coding RNAs stay associated with the chromatin, and then they serve as scaffolds to bind proteins, and those proteins that are involved in the process of action of the X chromosome called the bond. So could it be that there are RNAs involved here too? Sure. So this is a different, say, use of the idea of a scaffolding protein. This would be if we were to be clearer, maybe in the nomenclature, we'd have to say something like a chromosome structural scaffolding protein is what we're looking at here. So like the skeleton of it, which we know nothing about, but it's clearly there, right? You still got that X structure, so, and that's protein. And so we know that's there. This idea, I mean, it's certainly a related idea. But the scaffold is just in terms of binding particular other proteins, which is better understood at this point than the structure of the whole chromosome, because we know that we have particular binding sites on C300 that will bind particular binding sites on other transcription factors and coagulators holding in place. So at least here, we're seeing how the scaffolding works. So think of scaffolding in a broad sense. Scaffolding would be a scaffolding molecule would be any molecule that helps to organize complexes by means of binding different components at different places. So you've got like a binding site here for this, a binding site here for this, binding site here for this, binding site here for this. So you arrange these proteins sort of like a scaffold would arrange things as well, holding things in place. And when we talk about then, say, the scaffolding proteins that are used in structuring things like lipase chromosomes, the idea would be the same, that we have proteins that form like a whole scaffold for the whole chromosome.
Yeah, because in bacteria, they don't have histones really to organize their
Yeah.
DNA, so that's interesting.
Yeah, and they do have histone-like proteins. In other words, they have proteins that do bind to the DNA. So just like, and now, go to bacteria. I mean, I talk all the time about how we don't understand this yet, we don't understand this yet, and so when we're talking about eukaryote genetics, at this point, we understand eukaryote genetics at orders of magnitude better resolution than we understand bacterial genetics. So for a while, everything was about bacterial genetics, and that was where the fundamental models first came from. But the way they would study bacterial genetics is they would isolate particular regions of DNA, and they would put them in a culture with some RNA polymerases, and they would let them move down a track. Well, what you've done is you've destroyed the whole system. You've pulled isolated DNA out of the system, you've pulled an RNA polymerase, and you say, wow, we get transcription, so here's our model. RNA polymerase running like a train down a track. But you destroyed the system, and you still got some components that work, and you base your whole model on that, and that was, say, reapplied to eukaryote, and that's now the current model. But now we've been studying actual structure of how this works within eukaryote, and we know that model doesn't work. You never have a situation in